Hello everybody, this is going to be chapter 3 on cell structure and function, and I did upgrade my mic fairly recently, so hopefully the dogs that are 2 blocks away aren't too distracting during this recording. So this chapter deals a lot with the features of cells and how those cells can lead to growth, reproduction, responsiveness, and are responsible for their metabolism. So we're going to go through some of the different features of cells that allow them to do these 4 things. As we know, there are differences in the microbes that we are going to explore in this course. Some of them, of course, are biological or alive in that sense, and others are viruses. And so in this case, we'll talk about the features of cells exclusively, and we'll leave viruses for another lecture. So here we have some examples of cells. On the left, we have some bacteria. On the right, we have something a little bit more complex in terms of its external features, and we'll talk about how some of these differ. So when we're dealing with bacteria, we're typically dealing with cells that are extremely small, and we're talking about 1 micrometer in size. Eukaryotic cells, on the other hand, are slightly bigger, and they'll have some more ornate features, but we'll be able to take a look at some of those and what they look like. So like we all should know already, prokaryotes, they do not have a nucleus. They have instead a nucleoid region where most of their genetic information is kind of organized around. And one of the things that makes them a little bit different to eukaryotes is their ability to read and go through the process of protein synthesis. Whereas if you remember correctly from general biology, the DNA first has to be converted to messenger RNA, and then that RNA has to be processed, spliced, and then sent out of the nucleus into the different regions of the cell, including the rough ER, or in free-floating ribosomes in the cytoplasm, to be made into proteins. And of course, this is not something that bacterial cells need to do. They can go straight from that messenger right to the manufacturing of proteins. And so, yeah, they don't have the same limitations, and so they could do this fairly quickly. So here we have a very general picture of what a generalized prokaryotic cell would look like. As you can see, there are free-floating ribosomes just about everywhere. You have some inclusions, which we'll talk about a little bit later. They're just storage compartments within the cell. They all have to have a cell wall. They all have to have a cytoplasmic membrane. Some of them have flagella, like you see here. You also have the nucleoid region that I've discussed. And then on the outside, they'll also have the glycocalyx. And we'll talk about those as they're quite important for the survival of these organisms. In contrast to those prokaryotes, eukaryotes are a lot more complex. They have a nucleus, they have some membrane-bound organelles. They're typically larger in size, and these are anything like algae, protists, fungi, animal cells, plant cells, and we'll talk about those a little bit towards the end of the lecture. Here we have a very generalized animal cell. As you can see, the very, very clear nucleus, and you can kind of see that right there. You have nuclear pores, of course, as things need to enter and exit. Then you have a lot more complex things kind of on this side. You have lysosomes and the mitochondria, and then you have this little guy right there. So you see all those little dots. All those are ribosomes, part of the rough endoplasmic reticulum. And we'll talk about the endomembrane system as a whole as well. Here you have the smooth endoplasmic reticulum, and then you have the Golgi bodies. Those together, of course, making up the endomembrane system. And then we have some vesicles and all these other features that we'll also go into a little bit later. Here's just a comparison so that you can see the size differences. Of course, viruses are extremely small, not something that we were able to see until the transmission electron microscope. Then we have some bacteria, which we kind of knew existed for a while. And then, of course, you have other things like larger parasites that we can see. And they're all compared here against the chicken egg, which is something that we can all kind of find a reference to. Now, the glycocalyces are really important specifically for bacteria, because this is something that's kind of a feature that helps them survive. And so it's kind of this gelatinous substance outside of the cell, really similar to the extracellular matrix that you would find in some tissues of eukaryotic cells, or some eukaryotic cells. So these, however, are a little bit different. They're mainly composed of carbohydrates, proteins, and a couple of other substances. Definitely some moisture there, of course, and some fatty acids that help all of these things kind of aggregate into this mass. And these masses are eventually going to coat these bacteria cells, and they can do so in different ways. And hopefully we'll see an example on the next slide. And yes, actually here we have the capsule and the slime layer. The difference between capsules and slime layers is the organization of them. So capsules are typically repeating units of these different compounds or chemicals, and they're typically very firm and they attach to the surface of the cell quite well. It's kind of like a layer of protection versus a slime layer. It's a little bit looser. It is water soluble, but this is going to allow the bacteria to attach to surfaces, and typically whenever they find a food source, or they'll anchor to a place to live. And we'll talk about other features that are used for the same purpose as well. Here we have an image of what they look like. On the left, as you can see, it's a lot more uniform, it's a lot more dense, definitely there for protection. And then on the right, you see how it's a little bit looser, mainly for them to aggregate and to stick to different sources of food, for example. Now, flagella are one of the main mechanisms of movement for bacteria, and they're not very big. Their food sources are probably finite, and so wherever they can find them, they're going to try to either move to them, stick to them as quickly as possible. And so the flagella are paramount for them surviving, and so they can move these flagella in different ways, and we'll talk about the different arrangements of these flagella and how they move. Although they're not present in all bacteria, they are an extremely important part of their, I guess, anatomy. So they're composed of three main parts. You have the filament, which is the most visible part, the one that we all know. Then there's a hook, which anchors the basal body into the body of the bacteria, or the cell of the bacteria. So as you can see here, we have on the top left the gram-positive bacterial flagella. As you can see, there is the rod that kind of goes through that thick layer of peptidoglycan and the rings going and extending to the inside of the cytoplasm, with the hook and the filament kind of sticking out. On the bottom right, you can see the gram-negative structures. So as you can see, there's a few more rings within this structure that are helping it anchor that flagella into the cell wall and into the cell membrane of that cell. So there are different arrangements. Some of them could have a singular flagella, some of them can have multiple flagella, some of them can have the flagella on the exterior, some of them are a little bit more protected, but there's all these different varieties of moving around. So here we have the axial filament structures that are responsible for the movement of bacteria, like this spirochete right here. So as you can see, those filaments are found on one side of the bacteria and kind of go all the way around that, or all the way down that end. And so as these are contracting and moving, they're allowing the bacterium to kind of spiral almost, acting like a screw, embedding itself in to different things, including food sources or tissue in the case of an infection. So the function of flagella, of course, is to rotate, to help propel that bacteria through food source and environment, whatever it is that needs to get through. The rotation is reversible, so just like they can spin clockwise, they can also spin counterclockwise. And bacteria are going to move in response to the stimuli that they're going to see in that environment. So they're either going to try to go towards the food source or away from another sort of taxis or stimuli. And so these movements are known as the runs and the tumbles. As you can see right here, when we look at Paratrachus bacteria, when the flagella are moving in a clockwise orientation, this leads to the bacteria tumbling. So this allows them to change direction, and then when they're moving the flagella counterclockwise, they're actually running. And so the series of runs and tumbles is directed by the direction in which the bacteria are spinning their flagella. And so these are movements that a lot of these bacteria do in order to change direction, reorient themselves to be able to move wherever they need to go. Now, there are other structures like fimbria and pili that we need to consider. Fimbria are really important. They're kind of like bristles or little needles. If you think about like walking through a park and then you're wearing, I don't know why you would wear sandals with socks at a park, but if you were to do that, you'd probably come back to your car or to the sidewalk with a bunch of like, fuzzy things stuck to your clothing. And that's kind of what they do. They allow these bacteria to stick to different surfaces, and mainly food sources, or sticking to one another so that they'll be able to digest the food source whenever they find it. They're definitely a lot shorter than flagella, but they're really important for the aggregation and the secretion of some of those glycocalyces, which eventually are going to serve to develop what's known as the biofilm. And these biofilms are just these nice protective coverings that these organisms will produce to try to avoid detection by immune systems, or to try to be, I guess, to stick together so that they're not destroyed by water or many other different substances. And that's kind of what they look like. You see there'll be like little bristles. Those are the short ones that you see right here, pretty close to the cell. So eventually, of course, a bunch of them gather together, sticking together and secreting things. That's what's going to develop into the biofilm. And it's kind of like a little matrix or a little slime where those secretions will eventually aggregate together, kind of sealing them off from the outside. Now, pili are a little bit different. These are more with a purpose of conjugation, and conjugation is when two bacteria love each other and they want to exchange plasmids or DNA fragments. They'll come next to each other, they'll create the little tubes, and then once they attach to each other, they'll be able to exchange little bits of genetic information between one another. And so super, super useful. They can pick up some quite interesting features this way. And so here you have what the pilus would look like during bacterial conjugation. Now, in terms of their cell wall, they do differ. Typically, eukaryotic organisms will have cell walls in the form of cellulose or other compounds, but in bacteria specifically, we have gram-positive and gram-negative bacteria, and these bacterial cell walls are extremely important for the survival of these cells. The osmotic forces and pressures that are found within most environments are not the same than the interior that we have in bacterial cells. So, for example, when you take antibiotics, and these antibiotics are meant to disrupt the composition of the cell walls of bacteria, this now makes those bacterial cells susceptible to your body's osmotic pressures. And because of those differences, it sometimes leads to the death of these bacterial cells. And so this is really, really important, right? And not only that, being able to target specific bacterial cell walls so that you can treat them with the correct antibiotic is also really, really important. And so we need to understand the characteristics of what they look like and how they behave. In terms of their shapes, they're also quite different. And so you have, for example, on the top left in figure A, you have the coccus shape. This is a spherical bacteria. And when they're clustered like that, we call them staphylocultures. So on the bottom left under figure A, that would be a staphylococcus arrangement. And then you have also these chain-like shapes like you see towards the middle. And so because they are spherical in nature, but they are chain-like, this is a strep bacteria, so we call it a streptococcus bacteria. And then on the right, where you have those pink rods, this is a rod shape, so we call that a bacillus. And since they're clustered in a chain, we typically call it a streptobacillus bacteria. Now, these bacterial cell walls are made of peptidoglycan. That's going to be one of the biggest constituents of these cell walls. And they can be described as either gram-positive or gram-negative due to their staining capability. So comparing the traditional structure of glucose, which you have right there in that position, we have modifications to those. And the first modification is going to lead us to a chemical known as NAG, or N-acetylglucosamine. and you have just a different variant of a functional group in that position instead of the OH. And with NAM and acetylmuramic acid, you have a difference in the adjacent functional group right there. And so these two chemicals, when they're chained and they're alternated, they can do some pretty interesting things, including making it almost impossible for most enzymes to kind of seep in through those little spaces and break down those bonds. And so these bonds tend to be very, very durable, very sturdy, and they're very difficult to break. So here we kind of see how that arrangement looks like, where these alternating units are creating a lot of these different sheets, and these sheets are making it almost impossible for anything to get through into these bacterial cell walls. Now, the first type we're going to focus on is gram-positive bacteria. So gram-positive bacteria typically is going to stain purple after performing the gram stain, and they have a relatively thick layer of peptidoglycan. They have a lot of different types of chemicals, including teichoic acids and lipoteichoic acids, which are really, really important, as they are some of the things that allow for the resistance of bacteria to different compounds. And some of these, even like mycolic acids, making some types of bacteria very, very resistant to desiccation or to drying out. And I know maybe it's not as important here in the context of this chapter, but when we talk about things like identifying different strains of Mycobacterium tuberculosis, is going to be important to understand how that composition is important. And we will also talk about acid-fast bacteria during staining, or the chapter for staining, but just know that there are things that don't typically respond the same way to a traditional stain, and so you have to stain them a little bit differently, because in this case you won't be able to decolorize this bacteria like you would in a traditional gram stain. And so we'll talk about those processes a little bit later in the course. So here we have a little bit of that cell wall, as you can see right there, a very, very thick layer of peptidoglycan with some of these acids sticking out. Here we have that LPA. It typically goes all the way through. So in contrast to gram-positive bacteria, gram-negative bacteria have a very thin layer of peptidoglycan. In fact, this layer is fairly thin, and it is found in between two membranes. So you have an outer membrane and an inner membrane. The outer membrane, this is the one that contains several different things, including phospholipids, proteins, and a very important component known as lipopolysaccharide, or LPS. When bacteria sense that there's danger in the environment, or they're being exposed to your immune system or to antibiotics, and they begin to become decomposed or destroyed, they release an endotoxin known as lipid A. And lipid A is not something that's very nice to your system. It can cause a lot of different symptoms, including inflammation, fever. It can damage your blood vessels. You can have blood clotting, leukopenia, hypotension, eventually even things like septicemia and shock. And so it's really important when treating these infections that we're using, the appropriate amounts to avoid the release of some of these chemicals in excess, of course. And if you were to look at these under the microscope after a gram stain, they would appear pink. So here you kind of see what that looks like. As you can see, it's a little bit more complex in terms of the external structures, but nonetheless, both of these are quite important to the survival of these bacteria. Now, occasionally we do have bacteria that don't have cell walls, and there are a few of these, but sometimes they get mistaken by viruses. I mean, they're really tiny. Typically, they're almost perfectly spherical in shape. They do have ribosomes, they do have cell membranes, but a lot of the antibiotics that we use to make them susceptible to osmotic pressures, they really don't work here because there's no cell wall. And so what they do instead is they typically treat them with things like azithromycin that don't necessarily target cell walls directly, but cell membranes instead. So the cell membranes of these bacteria, they do have the same features that most other cell membranes have. They have integral proteins that span the length of the membrane, and then you have peripheral proteins which are found on the surface. And the best way to display this, if you remember from general biology, is the fluid mosaic model. And it just says that, these phospholipids are kind of not necessarily static, but they're able to move. And as they're moving around these large proteins that are embedded through it. And so here you can kind of see what that would look like with some of the things going all the way through. Of course, the phospholipids, so all of these regions are a little bit more flexible. So in terms of their function, the job of the cytoplasmic membrane is to regulate what enters and exits the cell. That's always going to be its primary function. In some bacteria, however, you have the capability of photosynthesis, and so in those cases, their job is to capture some of that light energy. So here in this case, we have an example where there is an electrical charge on that membrane, and as you can see, these cells will respond to the differences in changes in ions. Now, when it comes to transport is really similar. Like you have in a traditional cell, you have active transport and then you have passive transport. Some of the passive processes that occur in these cells are still diffusion, which is the movement of small particles across that membrane. You have facilitated diffusion, which still follows the concentration gradient, but there's channels for these compounds or these molecules. And then osmosis, which is typically facilitated with if the particles are small enough to fit through the membrane, of course they will. But you also have the assistance of things like aquaporins in some. So here you can kind of see how all those fit inside in the big picture. You could have some moving quite freely. Others require channels and things like that. So here we just have a simple demonstration of the osmotic pressure differences that can occur. Of course, water is always going to move across that membrane to try to balance or equalize the concentration from one side to the other. And so in some of these cases, like I said, that difference in pressure is going to make the bacterial cells burst, especially when the cell walls are not forming properly. And so here we have kind of the consequences of those differences in tonicity. Right? So whenever you have a cell that is equal to its environment in terms of salt concentration and osmotic pressure, you have what's known as an isotonic solution. So the cell will typically have no net movement. Water is still going to move in and out, but it's going to move in and out at equal rates. On the other hand, if you think of a child that is hyper, that means that they have too much energy. That's a similar thing that happens in a solution. If a solution is hypertonic, it means it has too much salt. And so in this case, there's going to be a net movement to the outside to where the solution is a lot more concentrated to try to equalize that concentration. In this case, you're going to cause the cell to shrivel up or to shrink in size. And when you have a hypotonic solution, that means that the concentration on the outside in this case is below the concentration on the inside. And therefore, water is going to move in to try to equalize the concentration on the inside, typically causing the cell to lyse or to burst, like you can see in this case. Now, active processes work a little bit differently. They don't necessarily go with the gradient in concentration. They typically oppose the concentration gradient. So instead of moving chemicals or ions along with the concentration gradient from high concentration to low, they will sometimes do it the opposite, from low to high. And so in some cases, these chemicals are not in the ideal concentration, and so the cell works to concentrate them so that it's able to carry out a lot of its metabolic processes. And some of these include translocations or modifications of these chemicals. So here we have an example of some of these mechanisms of active transport. You could have uniports that are designed to bring in molecules, and of course they're going to require energy because this is not going with the concentration gradient. Then you also have those that are antiport, well, where it brings in one type and it makes one type go out at the same time. Of course, this is still mediated by ATP. And then you also have coupled transport or symport, where you have the co-movement of these molecules. As one of them comes in, so does another. So in some cases, you also can have group translocation, like I mentioned previously, and this is when a substance is modified as it is moved within the cell. Oftentimes it takes energy from the molecule itself rather than ATP, but of course that's a topic for a later chapter, because it is an important part of something known as PTS, which is phosphatidyltransferase, which is a mechanism for some species of pathogenic bacteria, things like E. coli, Legionella, um, species among a few others. But again, this is one of the mechanisms in which the cell can directly kind of move the flagella or get the flagella to swim in a specific direction. Again, typically to follow the concentration gradient of something like sugars so that they can continue um, their metabolic processes. Here we have a table of the types of transport, table 3.2. I encourage you to examine this. It is a quite important topic for this chapter. So now the cytoplasm of bacteria. The cytosol, or the liquid jelly portion of the inside of the cell, is mostly water, and water is going to facilitate a lot of chemical reactions, so it's extremely important. But it also contains the DNA, and that DNA, of course, we've mentioned earlier, is found in the nucleoid region, which is where all that DNA is kind of clustered. You can also have inclusions, and those inclusions are typically deposits of different chemicals. In the case of a lot of bacteria, they'll actually store excess carbohydrates, and they'll do so in these little clusters known as the carbonosomes, as you can see, like in some of these that we have right here, which I think is quite interesting. And then we have the endospores, and endospores I find are quite interesting. You can typically stain some of these if you have a malachite green stain, and they appear quite nicely. And so one of the things that makes some species of bacteria so great at surviving is their ability to go into these forms that make them almost invulnerable to just about everything. And bacteria can sense their environment. They're not, just because they're simple life forms doesn't mean they're not complex and good at surviving. And so when they are exposed to specific stimuli from their environment, they'll go through the process of forming the endospore. And this endospore is resistant to a lot of different things. It can be resistant to heat or chemicals or just about anything you throw at it. And then later on, when it senses that there's no longer that threat or that harm, it'll come out of that form and it'll become biologically active again. And so here we have kind of the steps of how that works. So you have first the replication of DNA. And then as this DNA copy is made, you have kind of the separation of the interior of that cell. Half of that is eventually going to become the membrane of the cell and the second membrane of the cell. And as these membranes are being made, they're encapsulating one of the segments of DNA that was replicated, and they will create the cortex of the endospore. And then once that cortex is made, then the spore coat is going to be developed or hardened. And then as it develops and as it matures, it's going to use different things, including some of the chemicals that it took in, and through some processes that were really, really not known exactly or to that fine level of detail, they will eventually create the endospore. And the maturation of that endospore, like I said, it's not completely understood, but we do know that these can last for quite a very long time, and they're resistant to just about everything you throw at them. So here we have electron microscope picture of what these look like. Now, the cytoplasms of eukaryotes are slightly different. Of course, we have ribosomes in all cytoplasms, but in eukaryotes, the ribosomes that we have are slightly heavier. And in prokaryotes, they tend to be a little bit lighter. And all that number is telling us it's the sedimentation coefficient. And this is in Svedberg's units. So prokaryotes tend to have lighter ribosomes than eukaryotes. And so we can target that difference in size and cause changes only to one but not the other. That's how we're able to treat some organisms, by targeting the manufacturing of their proteins. Now, the cytoskeleton is really, really important in both prokaryotes and eukaryotes, as there's some of the things that will allow cells to divide, to maintain their shape, and to separate or compartmentalize different regions. For example, in the nucleoid region, they don't necessarily have a nucleus, but they're able to separate the DNA molecules from everything else. So here we have an example on how some of these structures can form the cytoskeleton of cells. Here you have a bioluminescent assay kind of showing you that helical shape of this cytoskeleton, for example. So some of the external structures of archaea, so these are a little bit different. Yes, they do have glycocalyces. They will develop into biofilms at some point, and they'll allow things to stick to each other and objects, of course, for food and other purposes, but still quite a very similar function. Flagella, also very, very similar, and they also have fimbriae, but they have something a little bit different. That's known as a hami. And this one kind of looks like a grappling hook, and if you were to look at some of these structures under the microscope, so here you can clearly see the shape of the hamus right there. It's very, very clearly resembling a grappling hook, so you can see that. And again, they're really good at attaching to things and holding on. So the cell walls of archaeans and bacteria are different, very, very different. They don't have typical peptidoglycan. They have different things that they use for their cell walls. Some of them, of course, being a chemical compound known as pseudopeptidoglycan, which I'm not sure if your textbook discusses in a lot of depth, but definitely important differences there. They have a different composition, and typically antibiotics that are really, really good at dealing with bacteria really don't do much for these types of prokaryotic organisms. So we kind of see some of those shapes and different morphologies of prokaryotic organism. Again, something that you typically don't see in other prokaryotes like bacteria. Now, the cytoplasm, it's still really important for the maintenance of chemical reactions. You do have some similarities in the ribosomes, the cytoskeleton, even the organization of the DNA structures. However, some of the proteins are slightly different and slightly modified, specifically when it relates to the metabolism of some of the enzymes that make it. And we're talking about the polymerases and that type of enzyme that we see. And then you also have some similarities in the genetic codes in terms of the overlapping, some of the things that are used for processing of the information. Here we can kind of see some of the features and how they differ in both of these groups of prokaryotes. So it's an important part that I encourage you to look at. Now, in eukaryotic cell, glycocalyces are a little bit different. They're not as organized and as dense as you would see them in capsules of prokaryotic organisms by a long shot. The extracellular matrix in eukaryotic cells is definitely different. Animal cells specifically have to communicate with each other, and any multicellular organism for that matter has to communicate with one another. And so the recognition of these cells to the self and the communication between these cell types is extremely important. Not only that, but the availability of nutrients and things like moisture, and the ability of cells just to stick together in general is one of the main importances of the glycocalyces. A lot of cells don't have cell walls. There are fungi, algae, plants, animals, protozoans that don't really have that there. And if they do, sometimes they're modified. So for example, you can have plants that have cellulose making up their cell wall. You could have fungi that have chitin, cellulose glucomannan, which is a different compound. Um, you can have protein compounds that are also making up these cell walls. So there's a lot of diversity when it comes to eukaryotic cells and their cell walls. In terms of membranes, however, they all do share similarities. So here we have a eukaryotic cell wall. Here, I'm not sure what we are looking at, but as you can see, that cell wall is pretty thick, and the membranes are fairly small compared to them, or right next to them. There are cytoplasmic membranes in all eukaryotic cells, and again, the same fluid mosaic model where you can have integral or peripheral proteins still apply. In this case, you do have a lot more steroids that are embedded within these membranes. Their main purpose is for fluidity. One of the things that you have with eukaryotic organisms is multicellularity, and in some cases, multicellularity comes with movement. And so in this case, you want to make sure that those cells are fluid, they're able to change and shift. And so cholesterol can help those cells survive some of those stressors. There are regions that you have that are known as membrane rafts, where you have several lipids and proteins kind of clustered together. But we'll talk about that probably at a later time. But there are things that also can help things move in and out of the cell, and these cell membranes are largely responsible for that. So here you have two cytoplasmic membranes and an intercellular matrix kind of in between them, helping them communicate and anchor themselves to each other. So here we can kind of visualize the process of endocytosis with this amoeba. So this amoeba is taking in another cell, kind of looks like a euglenoid. And so as it takes it in, the membrane of the cell is going to create a vesicle for the thing that it has ingested, and then eventually at some point on the inside it's going to release digestive enzymes or other chemical compounds to degrade this and have it as food. So make sure you take a look at table 3.4 in your text as it describes some of the processes like endocytosis and exocytosis. Now, when it comes to flagella in eukaryotes, they function differently than they do in bacteria, not only in their composition, but in the movement itself. So in terms of their structure, they're actually held together by the basal body and not the hook like we see in bacteria. You could still have single or multiple, but typically they're going to be polar. So they're going to be at the poles and not throughout the body like you have in some types of bacteria. So here we have kind of what that would look like. So here we can clearly see the differences in the structure of the internal workings of the flagella. So on the outside of the body of the cell, you have the 9+2 arrangements where you have a pair of microtubules in the center surrounded by 9 bundles of those pairs. Whereas towards the inside of the cell, you have the 9+0. So there's a transition where now you have 9 bundles of groups of 3 with a 0 in the middle instead. So they function really similarly. They're both mechanisms of movement. However, in contrast to the bacteria's rotating movement, these are a little bit more rhythmic. So instead of rotating in circles, these kind of go up and down or back and forth. And as they do that, they create a webbing motion that helps propel them through the medium or through body of water, whatever it is. So here you can kind of see that that's a lot more rhythmic than you have compared to the rotation of the bacterial flagella. Now, cilia are definitely a lot shorter than flagella, and they're just coordinated little hairs that are going to move typically the cell through the environment, not necessarily against a current. I don't think they're that strong, but definitely they can orient the cell into a more favorable position so that they can kind of move a little bit better. But they're also used to move kind of things over them, especially if they need to target them to a specific place, including things like their mouths. And this is kind of what they would look like. Of course, flagella being much longer than that. And cilia, they have a little bit more freedom when it comes to their movement. Of course, they can still rotate and move slightly in different directions, whereas flagella are a little bit more back and forth. So other organelles include the ribosomes. Of course, these ribosomes are a lot heavier and larger than you would have in prokaryotic organisms. And the cytoskeleton is a little bit different as well. So in this case, they help keep the organelles in place, and they create this extensive network of fibers and tubes that are moving things within the cell. And so when we think about cells and when they manufacture proteins, sometimes people just think that, well, the cells make these proteins in the ribosomes, and then somehow magically these products or proteins just float their way to wherever it is they need to go. And the reality is that that doesn't happen. In order for those proteins to be taken somewhere, they have to be encapsulated, and then they have to be tagged. And this typically happens through the endomembrane system. And then once they're packaged into the vesicle, there are little proteins, they're called kinesins, that will move these vesicles from one end of the cell to another, wherever the target is, across these little tracks, and those tracks are the microtubules that are part of the cytoskeleton. And so all these things, they're super, super important and valuable to these cells. And as these cells become more and more complex, they're going to require some sort of infrastructure to allow all these changes and these chemical reactions to take place separately and be able to deliver things to where they need to go. Okay? So here we have kind of what that is made of. So these are microtubules on the left, the ones in green, and then on the right you have actin filaments. You can also have protein intermediate filaments that are also used, especially when it comes to like division of cells and stuff like that. But definitely something that you wouldn't typically see in bacterial cells. Now, centrioles and centrosomes are not a universal component of eukaryotic cells. Typically, you might not see these in plants, for example. But they're definitely extremely important for things like cell division and cytokinesis, the development of flagella, cilia, and some of these other filamentous components of cells. But this is kind of what they look like. Of course, these tubules are going to grow in specific directions as they're stimulated by different processes within the cell. And then we have now some of the membranous organelles. And the nucleus is, something that everybody remembers, that they all know that all, animal cells and plant cells and eukaryotic cells have a nucleus. And yes, they all do. Inside of the nucleus, you have the nucleoplasm, which is kind of the analogy to the cytoplasm of a cell. And inside of this nucleoplasm is where you have your chromatin. So this is when the DNA is found kind of in an uncondensed form. When it's time to go through the process of replication, then this chromatin will condense on these little proteins called histones, and then those histones will eventually form what everybody knows and loves as the chromosome, right? And then these chromosomes will eventually go through the process of division, right? But until then, that's where they're found. RNA is synthesized in the nucleolus, which is found inside of the nucleoplasm. And this entire thing is surrounded by a nuclear envelope that has nuclear pores. And of course, this is a very VIP area, very exclusive. The only way to get in there is if you have something known as an NLS signal, the nuclear localization signal, which will allow things to be delivered into the nucleus. And then, of course, once it's done, it has to make its way out. So here is an image of what that would look like. As you can see, there's quite an extensive network of those pores and folds in these membranes. It's just quite interesting. Now the endoplasmic reticulum is the next part. So this is just a bunch of flattened sacs that have a lumen or a space in the middle that is going to allow for the synthesis of things like proteins, but as well as a transport system to get things from point A to point B. So you have two versions of the endoplasmic reticulum. You have the smooth, which has no ribosomes, and then you have the rough, which has ribosomes. And the ribosomes are what kind of give it its little studded appearance, like it's rough. So here you can kind of see both of them right next to each other. And now the Golgi body or the Golgi apparatus, this is where you have your secretory vesicles. And this is kind of where they would go into their desired location, or if need be, they will fuse to the cytoplasmic membrane and then be released to the exterior of the cell. Of course, this is not something that happens in all eukaryotic cells, but definitely worth a mention. This is kind of what it looks like. I think it's pretty cool. Now you have other organelles like the lysosomes and the peroxisomes, as well as the vacuole and the vesicles. A lot of these things are used to store chemicals, transfer chemicals, but in some of these, for example, your lysosomes and your peroxisomes, they're a little bit more metabolically active. So lysosomes, for example, they have catabolic enzymes, which are responsible for the breakdown of different substances within the cell. And the peroxisomes in this case, they're there to degrade potentially poisonous waste that is produced as a byproduct of cell metabolism. Here we can see the vacuole. This is something that we have in plants and some protists. And you can see it takes up a large amount of space, and there's different reasons for those. Some of them can be stored or filled with pigments, others with water to maintain things like turgor pressure, salts, other things, because they're not readily able to just secrete things out in vesicles like an animal cell, for example. They would have to store these. And again, in some cases, maybe you're looking at the leaves of a plant. Eventually, like if you have a plant that's able to uptake salts, those would be stored there. And then when the leaves fall, they would be redeposited into the soil, for example. And so there's a lot of different important functions for these. So here we have the role of vesicles in endo and exocytosis. So let's say that the immune cell finds a bacteria floating around that could be potentially dangerous. This cell will phagocytize that bacterium, and so it'll create a little vesicle around it like we see here. And then if it identifies that it is potentially hazardous, it'll send a lysosome. The lysosome will fuse with that vesicle, then destroy it, whatever's inside that vesicle, through its digestive enzymes, and then eventually release it back. And when we talk about immunology, we'll talk about the process that actually happens within an immune cell. Really similar to this, but with a few extra steps. But until then, this is a very simple process that I think does the job. Then we have the mitochondria. So I'm sure everybody is sick of hearing that the mitochondria is the powerhouse of the cell. So I know this probably won't catch on, but we could say the mitochondria is similar to alpha proteobacteria. And hopefully you'll get that by the end of the semester. But anyway, it does have a double membrane, which is consistent with the endosymbiosis theory, which we'll talk about in a couple more slides. But this is where most of the ATP is going to be produced by every living thing. We do have some substrate level phosphorylation that will produce some ATP, but predominantly the mitochondria is where most of this energy is coming from. And so you have an interior matrix that has its own ribosomes, that has its own circular DNA, and it's able to go through the process of division without the regulation of the nucleus, which I think is pretty interesting. It's kind of like you had a little bacteria in there that was doing whatever it wanted, and then it became dependent on the larger cell. And that's kind of what the endosymbiosis theory kind of says, is, at some point there might have been a relationship between these two, but then one of them lost the ability to replicate freely without the other. And so therefore, over millions of years, these two became dependent. And so therefore, now they must grow one with the other. And so here we have that that traditional mitochondrial shape, as you can see, and you have all those little folds on the inside. Of course, all of those folds are there for an increase in surface area so that you're able to keep up with the ATP production required by cells. And the chloroplast is, if something happens and it's great, it probably will happen again. Now, the chloroplast has a really similar function in terms of not necessarily producing ATP, because although it can produce ATP in small quantities, its job is mainly to synthesize glucose and capture some of that light energy to create that glucose. Um, but I'm sure we'll talk about the process in photosynthetic bacteria. But here, of course, you have your own ribosomes, you have your own DNA, and they're able to do very, very similar processes. However, in contrast to mitochondria, this chloroplast, it has quadruple membranes rather than just a double membrane. And so it tells us a little bit more about the evolutionary history of this specific organelle. And so here you have kind of what it looks like. And so the basic structure here um, we have the thylakoid, which is, if you were to think of these as pancakes, the thylakoid would be an independent pancake. The grana or the granum, this would be a stack of pancakes. And then the stroma would be the syrup that covers most of these things, right? And so this is kind of what that would look like. So I would make sure to look at table 3.5 as it includes both membranous and not membranous organelles, their general function, and their presence in prokaryotic and eukaryotic organisms. And it does span 2 slides, so I encourage you to look at both. And of course, definitely not least is the endosymbiosis theory. So of course, that tells us about the union of aerobic bacteria with larger anaerobic prokaryotes, mainly those that were able to oxidize pyruvate and process pyruvate even further. And as they were processing pyruvate, they were able to maximize the energy outputs versus doing this by themselves. Of course, smaller prokaryotes were the ones that eventually became the parasites, and then at some point that independence or the existence independently made its way out, and the larger cell was also dependent on the small cell to survive or for ATP production purposes. And so eventually these bacterium or organisms that might have resembled bacterium became turned into or became the mitochondria and the chloroplasts. And so over millions of years, these relationships have survived. And so all eukaryotic organisms, for example, they all contain mitochondria, even if they're plants. And I know when we're in grade school, we're taught that, oh, well, a plant could just stand out in the sun and it could get energy from the sun. And the reality is, yeah, they get energy to make a little bit of ATP and glucose, but glucose still has to be broken down by the cell. And in this case, yes, plants do have mitochondria, and they break down the sugar that they make during photosynthesis. And so mitochondria is one of the most important features of eukaryotic cells, as it is the powerhouse of the cell. Make sure to take a look at table 3.6 as it compares these cells and their features.